Hello. Well, thank you very much for joining us in this Genomics England seminar, today. And in fact, we've got two excellent speakers to speak about their work, and on the pan-cancer area, right. So if I could just, it's firstly, I'm Richard Holston. I'm one of the people are helping GEL along with my colleagues Anna Shu and Matt Lerner, involved in was trying to coordinate some activity with respect to the Pan-Cancer and Molecular Oncology community, which is in fact is one of the 8 communities within the GEL system, right? Before we actually hear anything from the speakers, then I'll just try and update those that are not very familiar with the GEL system and experience, just to give you an idea of the terms of reference and actually how exciting it is as an initiative. May I just make a couple of points though? Firstly, that this is actually recorded, right, and it will appear on YouTube. So if you actually miss any of these activities, then you can catch them all on YouTube, right. There'll be 20-minute presentations. The two speakers who have kindly given their time to speak, followed by 5 minutes of audience questioning, and then I'll just be wrapping it up at the end, right. So as I said, there are 8 themes within the GEL system, and the pan-cancer molecular oncology area is in fact one of the largest, within the communities. We've got about over 530 members, and we've actually currently there are about over 270 projects that are active. It's about the 4th largest thing. So it covers all the aspects, the projects, from fundamental discovery projects to identification of therapeutic targets, identification of biomarkers, and also the exploration of new technologies for clinical value and expanding cancer across the whole board. Right. So as I mentioned, I'm one of the 3 people helping the Genomics England team who have got lots of expertise. There's Mana Shu, who was actually instrumental in actually setting up the 100,000 genome project along with Matt Caulfield, etc. And then we've most likely recently been joined by Matt Lecter from UCL, whose main interest is in head and neck cancers. Right. So what we're trying to promote by these is to actually increase the idea of increase the opportunities for networking and knowledge sharing, bringing together collaborations in different research areas. And also, we obviously want to champion engagement of early career researchers and participant representation, identify and basically communicate the research, and hopefully address the issues about maximizing funding opportunities as well. So in Genomics England actually has got a large ambition for cancer research, supporting over early diagnosis and treatment through research and personalized therapies, translating these for clinical benefit, and also generating multi-omic data sets for discovery projects, in the National Genomic Research Library, right. So I just want to touch on the 3 point. So it's gone through a number of incarnations, and development as it's progressed since its early inception. And in Cancer 3.0, there's going to be more efforts for clinical trial matching, deep multi-omic analysis that was going to obviously facilitate early detection and better diagnosis. Right. So I'd like to just make an announcement before we go into the presentations. There's the thank you all for joining the seminar today. There's going to be a presentation at Genomics England presentation at the ESHG in the 26th of May. There's the Genomics England Summit coming up in June, at the Business Design Centre, and registration is currently open. If you have any questions about ongoing activity within the GEL system or anything, you can always contact the research management team who give prompt responses, right. So there's an opportunity for currently within the NGLL to basically get much more information on the pathology data, which is now going to be one of the significant thrust of GEL in going forward, to actually produce better information for multimodal analyses. Right. So, and this is open to development, and obviously once encouraging people to take advantage of the opportunity within the UK. So, in fact, if I'd like to just introduce our first speaker, who is Chris Bailey, who's going to talk about the origins and clinical impact of extrachromosomal DNA across the 39 tumor types. And Chris is actually a clinical research fellow at UCL with Charlie working closely with Charlie Swanton, and then he'll be followed afterwards by Daniella Black, who's been working with Sabrina Xaney. Insights from breast cancer, whole genome data linked to mortality. So I'll hand you over to Chris to kick off the show, if I may. Thank you, Chris.
Thank you, Richard. I'm just going to see that I can share my slides. Um, is that working and on the correct view?
I think you need to go bigger, basically.
Uh,
It's all good.
all good. Okay, excellent. So, yeah, I'd like to start by thanking Richard and the organizers for giving me the opportunity to talk about extrachromosomal DNA and the project that we as a team completed in the Genomics England environment. So for this talk, I'm going to briefly outline what ecDNA is and how we detect them to those that are not familiar with the phenomenon. I'll talk a bit about the origin and function of ecDNA across multiple tumor types. Um, and then how we have inferred mutational processes and underlying genome instability surrounding ecDNA formation and maintenance. And then finally, the clinical implications of ecDNA from the analysis within the Genomics England environment. So I'll start by talking about what ecDNA is and how we detect them. Just to say that over the last 5 years, ecDNA has sort of captured the public imagination, has been sort of referenced multiple times in the broadsheet. So this is a headline from The Guardian in 2023, which has sort of called ecDNA this Bond villain DNA, which could transform cancer treatments. And another in the New York Times from 2019 that they have sort of claimed that scientists are just beginning to understand mysterious DNA circles that are common in cancer cells. So quite a lot of interest, quite a lot of imagination that has been captured. But what is ecDNA? So ecDNA are circular structures of 2 to 5 megabases that often contain identical copies of genes and reside outside conventional chromosomes. They create focal copy number amplifications. And prior to next generation sequencing, they were traditionally identified on chromosomal based cytogenetic preparations, such as metaphase spreads. So, traditionally, focal copy number amplifications are characterized as intrachromosomal. So, what you can see here, hopefully, my mouse is clear, but you're seeing in pink this sort of MYC hybridization and this cluster of MYC genes on an intrachromosomally within the spread. So these are called homogeneous staining regions or HSRs. And at the bottom here, again, the metaphase spread, you see these MYC probes hybridizing to pieces of DNA outside of the chromosomes themselves. So these were traditionally coined HSRs or double minute or ecDNA, and you can sort of see the difference between them within these cytogenetic preparations. So one of the things ecDNA does is it generates ultra-high copy number amplifications, and this is due to the non-Mendelian inheritance pattern. It happens because ecDNA do not contain centromeres, and this creates a random segregation event which can amplify the number of copies of genes within successive cell divisions. So in this cartoon, what you normally get is a cell when it divides, you get an equal amount of genetic material in the daughter cells. But because of this random segregation, in this schematic, after 4 successive generations, you get 9 copies or 9 ecDNA with a single gene copy on each ecDNA. Whereas normally what would happen is that you would have the same number of genes intrachromosomally that are transferred within each successive cell division. So you would normally get 2 copies of the genes here instead of 9 copies. This cell-to-cell variation exacerbates intratumor heterogeneity and creates the sort of fuel for which tumor ovulation can occur. We know that ecDNA congregates in dynamic hubs, and this permits this trans interaction and co-option of regulatory regions that boosts transcription. And in 2022, King Hung and colleagues showed that the application of JQ1, which is a BRID4 inhibitor, disperses these ecDNA hubs. And that is one of many lines of inquiry around how we can target tumors that specifically contain ecDNA. There have been a number of examples of ecDNA-driven drug resistance, and I'll touch on 3 here. So one was published by Nathanson et al. As part of Paul Mitchell's lab in 2016 in Science, where they had EGFR V3, commonly found in glioblastoma. These amplified clones become resistant to alotinib through homogeneous staining region formation. So you have this preparation, which is naive to alotinib. On the application of alotinib, you get this HSR formation, so these alotinib resistance, which have occurred through selection. And then once the drug is removed, again, it's thought through selection, you get this re-emergence of ecDNA containing clones. Another model of ecDNA-driven drug resistance, which was published by Ofer Shoshani and colleagues in Don Cleveland's lab, was this induction of dihydrofolate reductase amplification, which had formed on ecDNA as a consequence of chromothripsis. And this experiment occurred through the application of methotrexate at increasing concentrations on a population of HeLa clones, and as the increase of methotrexate, the increase in concentration of methotrexate was occurred, there were higher and higher copies of dihydrofolate reductase, which is the target of methotrexate. And so these resistant clones at increased concentrations had ecDNA formed through chromothriptic events, and these were the drug-resistant phenotypes. We don't just see this in human cell lines, but these are seen in plants as well. So this, again, high-level amplification has been observed in Amaranthus palmeri, which is pigweed, and glyphosate-resistant Amaranthus palmeri contains ecDNA with the EPS-PS gene. And EPS-PS is the target of glyphosate in the Amaranthus palmeri. And a paper published this month has shown that a new ecDNA has been described, which is resistant to both glyphosate and glufosinate ammonium. So this is a mechanism of resistance that occurs not just in humans, but has occurred in plant biology as well. So with whole genome sequencing data, we can infer ecDNA through computational tools. And in this particular study, we took whole genome sequencing from Genomics England. We aligned to the reference genome. We used amplifications, focal amplification signals as seed intervals for amplicon architect analysis. And then we used a tool called Amplicon Architect, developed by colleagues Paul Mitchell and Vineet Bafna, to determine ecDNA or focal intrachromal amplifications based on their content. Amplicon classifier then identifies these focal amplifications and features them into cycles or paths. For a number of samples, we were able to determine cytogenetically the pattern of amplification. And so two examples you see here. So on the left, This is an intrachromosomal amplification of PDGFR alpha and an extraskeletal osteosarcoma. Much like the previous slide when I showed you the metaphase spread and the HSR formation, this kind of clumping of gene copies. This is seen here in this predicted intrachromosomal PDGFRA amplification. And then we have the MDM2 CDK4 ecDNA shown here, and this is an interface fish of a dedifferentiated liposarcoma. So that's ecDNA and how we detect them. So the origin and function of ecDNA across multiple tumor types. So we use the Genomics England V12 cohort, and after a number of filtering steps, we got to 14,778 cancer patients of 39 different tumor types, and the whole genome sequencing data is of 100x tumor and 30x germline normal, both of which were used to make these computational predictions. So the prevalence of ecDNA across 39 tumor types came to a cohort level estimate of 17.1%. We used the stage breakdown for our multivariable models, but the highest proportion of ecDNA were found within the liposarcomas, glioblastomas, and HER2 breast cancer samples. We found that the oncogenic content of ecDNA was higher than chromosomal amplifications, and we found that genes recurrently encoded by ecDNA were more likely to be oncogenes compared with non-ecDNA driven vocal amplifications. We found EGFR and ERBB2 as not only commonly amplified, but they were commonly mutated, and those mutations tended to be under positive selection. So a good example is of EGFR, found 130 times in the cohort, and within the EGFR gene, when they were also amplified, they tended to be co-mutated. And running a DNDS analysis within these focal amplifications, we found that there was a signal of positive selection for EGFR and MDM2 when they were amplified versus when they were not amplified, taking all of the mutations within the gene itself. So what about ecDNA that do not contain oncogenes? So we saw that traditionally most ecDNA tended to amplify, traditionally it was known that the purpose of ecDNA was to amplify oncogenes. And we saw that in most cases when we took the Bushman annotation, we found that 65% of ecDNA contained oncogenes. However, when we removed those ecDNA and then we asked, well, what is the proportion of ecDNA that do not contain oncogenes, we found that a little under 50% of the ecDNA, taking into account the gene content and removing those ecDNA that contain oncogenes, a large proportion of those genes, those ecDNA contained immunomodulatory genes, and in some cases, we were only able to annotate regulatory elements onto those ecDNA themselves. So when we took the non-oncogenic ecDNA and we ran an overrepresentation analysis, we found that there was an enrichment for genes that were involved in immunomodulation. So particularly natural killer cell mediated cytotoxicity, negative regulation of lymphocyte mediated immunity, and natural killer cell mediated immunity. We then asked and had developed a tool within the lab that allowed us to estimate the T cell fraction of a tumor using whole genome sequencing. We then asked, is there a fundamental difference between those samples with immunomodulatory genes versus those ecDNA with immunomodulatory genes versus those without. And we found that there was a depletion of T cell content within those genes that contained immunomodulatory, those ecDNA that contain immunomodulatory genes. So how might immunomodulatory ecDNA work? Well, we thought potentially two hypotheses. One is that a cancer cell may amplify a gene that attenuates NK cell mediated killing. So for example, amplification of HLA-G. Another thought is that the cancer cells may hijack proteins that are normally found on regulatory T cells that downregulate cytotoxic lymphocytes, for example, LRCC32. So then the next part was to infer mutational processes and underlying genome instability surrounding ecDNA formation and maintenance. So the first thing that we did was to compare those ecDNA, those samples with ecDNA and those samples with focal amplifications, and annotate whether or not there were tumor suppressor losses, and then conducted a multivariate analysis looking at the presence or absence of a TP50 of a tumor suppressor mutation versus those that did not have the mutation, and we found that TP53 was most strongly associated with ecDNA presence. Interestingly, when we subsetted by MDM2 ecDNA only, we found a mutual exclusivity between TP53 status and MDM2 ecDNA status. So those samples that had MDM2 ecDNA were TP53 negative, despite the fact that across the cohort, TP53 mutations were most strongly associated with ecDNA presence. We were then able to conduct what we call an amplicon entropy score. We were able to calculate an amplicon entropy score, which essentially took into context the number of structural variants and copy number shifts within a focal amplification. And so those samples with high entropy tended to be more complex ecDNA, and those that only had one or two structural variant breakpoints with no or very few copy number shifts tended to be low entropy. And when we stratified this by tumor type, we found that sarcomas contain highly complex ecDNA related to increased structural variant burden, whilst colorectal tumors tended to have non-complex, thought to be episomal ecDNA, more related to underlying genome instability and whole genome doubling. So sarcomas highly enriched in structural variants and complex DNA, complex ecDNA, known to be highly have high prevalence of chromothripsis, tended to form these very complex ecDNA, whereas colorectal tumors were tended to be more episomal and related more strongly to whole genome doubling and whole genome weighted genome instability. Then following a collaboration with Serena Nick Zainal and the science paper published, I think it was about 3 years ago now, by Andrea Di Gasperi, we used some of the analysis of SBS signatures within the same samples that were conducted using the same data set, and we were able to create a model that looked at the enrichment of certain SBS signatures amongst the samples that contained ecDNA. And we found that there was an enrichment for SBS4, SBS13, and SBS8 within samples that had ecDNA. And there was a negative association with mismatch repair deficiency. As we increased, we looked at hypermutated samples, and we found that there was a negative association between hypermutation, whether it was related to POL-E or POL-D1, or MMRD, there was a negative association with hypermutation as a consequence of these mutational processes and ecDNA presence. We were then able to time mutational signatures, SBS processes, and we were able to annotate whether or not these processes occurred pre or post ecDNA formation. We were able to do that by taking the variant allele frequency of the mutations within the ecDNA regions. We were able to get a mutational copy. And if the ecDNA copy matched the mutational copy, these were inferred as occurring pre-ecDNA formation. And in those where the mutational copies, according to the variant allele frequency, were less than the total copies of ecDNA, they were classed as late. And we found that SPS3 was enriched post ecDNA formation, whereas SPS4, 1, and 92 were enriched early, so it occurred pre-ecDNA formation. We then were able to take the clinical annotation within the Genomics England data sets. I think one of the strengths of the data set in general was being able to overlay some of the survival data and some of the treatment data. We found that ecDNA was associated with metastasis and later tumor stage, and that the detection of ecDNA was associated with poor survival. We also found that in a multivariable model that the presence of ecDNA was associated with poor survival, even when controlled for stage, age, and perhaps most strikingly, underlying genome instability. So one of the original thoughts was the reason that ecDNA results in poor survival is because the tumor is underlying the tumor has some underlying genome instability, and that inherent genome instability is the thing that that contributes the most to poor survival in the ecDNA context. But we attempted to control for this, and we found that ecDNA was still associated with poor survival despite that. We then also found that in those samples that were pre-treated, that there was an enrichment for ecDNA presence, again when controlled for stage. And that sort of concluded the study itself. So in conclusion to this talk, we described the prevalence of ecDNA across 39 tumor types. We identify possible novel functions of ecDNA, such as immunomodulation. We demonstrate that ecDNA has multiple underlying mechanisms that are dependent on underlying tumor type and show that ecDNA is associated with late-stage metastatic disease, treatment, and poorer overall outcomes. And there are a couple of avenues that we are hoping to look at. One of these is differential methylation. So we have found that within ecDNA, there are specific unmethylated CpGs at the locus of the oncogene itself, and that the rest of the ecDNA is effectively methylated and sort of switched off. We'd like to know that understand whether these non-oncogenic ecDNA behave in the same way. And we want to dig in a bit more into this HER2 heterogeneity and trastuzumab resistance. It's been shown that the HER2 heterogeneity correlates with treatment resistance, copy number heterogeneity. And it would be interesting to see whether this copy number heterogeneity is driven by ecDNA or not. So I'd like to thank many people who contributed to this particular study. The grand challenge in particular, so Paul, Vineet, Howard, Serena, King, Jens, Ben, and Natasha. The GSIP collaborators, so Alex Cornish was incredibly helpful in providing some data for us. And then Charlie Swanton, Nikki McGranahan, and Mariam Jamal Hanjani as well. Okay, and that's me done. Thank you.
Thank you, Chris. That was an excellent overview of a very complicated subject. So I think we're probably going to have some. I think we can you see the question and answers? Sorry, the Q&A. Have you lead that already? And perhaps you'd
Yeah.
like to give an answer. Sorry.
Yes, so ecDNA seem to be more prevalent in tumor types that often undergo ALT telomerase stabilization. Is there any commonality of mechanism, e.g., ATRX mutation or predisposition? Do you see telomere circles in the same samples? That is an excellent question. So it's something that we've been working on with Simon Bolton's lab. Who is very interested in ALT and these sort of T circles that are that maintain telomere length. The computational tool does not predict very well the telomeric ecDNA just because of the sequence nature of the telomeres. But one of the things that we are doing at the moment is trying to identify whether the genes that correlate with ALT formation are seen or enriched in ecDNA formation as well. And we have some cell lines on the go as well. So we're performing screens to understand whether those genes that are implicated in ALT are also implicated in ecDNA formation. So we haven't done it yet, but there is a natural synergy between the two processes, and we think we'll find some commonality.
We've actually got a feast of other questions coming in. If you can manage those within your allotted time, it'd be very good.
Yeah, so can ecDNA activate the immune system at brain cytoplasm after mitosis? So we think that's a very good question. So we are looking at the grand challenge is looking at whether ecDNA can activate the cGAS sting pathway. And there is evidence to suggest that there is cGAS sting activation even especially when the ecDNA is cytosolic. I think it's something that again is under intense investigation, and I think I would suggest I would uh, postulate that this is true, that ecDNA can activate the innate immune system if it remains in the cytoplasm. So we think yes, we think this is cGAS sting, but it's something under investigation at the moment. What advantages do you think ecDNA confer towards tumor survival? So I think there's a few things. So one is we know that uh, there are specific ecDNA driven mechanisms of tumor of drug resistance, of targeted therapy resistance, which have been demonstrated in vivo. And we know that the genes that are amplified by ecDNA are kind of spread across the genome. You focus on oncogenes that are amplified, but actually there are a variety of other genes that are also amplified, and we don't know what they're doing. But given that we've seen ecDNA-driven mechanisms of drug resistance around amplifications, I would say that that was one thing that conferred tumor survival. I think the other thing that's quite apparent is that ecDNA fuels intratumor heterogeneity, and I think that intratumor heterogeneity in itself is a mechanism that allows the tumor to gain survival advantages through cancer evolution. So they are the two things that I think are most pertinent in terms of ecDNA's ability to tumor survival. The other thing is that I think gene amplification in itself is something that promotes growth and survival advantages because you're sort of supercharging these oncogenes. So I think that's a third thing. Do you think the presence of immunomodulatory ecDNA is an early event? There's evidence in the esophageal cancers that these circles. Yeah, that's true. I think it's the same for in breast cancer as well, carcinoma in situ, and we are looking at the same thing in lung as well. We haven't tested whether or not this is an early event, and I find computationally inferring ecDNA presence, we normally build phylogenetic trees based on mutational clustering. And because ecDNA doesn't quite obey the rules of Mendelian inheritance, being able to time ecDNA is quite tricky. I think the studies that will show this will be those where you can get samples, pre-invasive samples. But I think it's a neat thought that immunomodulatory ecDNA is an early event. You get this kind of immune escape, and then you have sort of cancer initiation from there. So I think it's a neat theory. I think to answer it properly, we either need to be better at our computational phylogenies involving ecDNA, could be done with structural variants, or we get samples at an earlier time. Technical question. L-carnitine uses WNFD since they're carbonic sequence in the first place and detected normally if WGS doesn't have a restriction digestion step. Maybe I'm missing something. Yeah, so if yeah, so these cycles are quite big. So these are sort of mega-based sized circles. So without it, we get shear, and this shear kind of fragments the ecDNA, and so WGS captures these much larger circles because of the shear that's involved in the WGS preparation. I think the smaller ecc-DNA do require restriction digestion. So smaller ecc-DNA are hundreds of bases to thousands of bases as opposed to millions of bases. And so it's much harder to detect ecc-DNA using amplicon architect because these aren't necessarily captured in WGS. They can be captured if you were to use a plasmid safe ecDNA exonuclease preparation. So you remove the linear DNA and then you perform restriction and digestion, and that sort of enriches for these ecDNA. But the larger circles, you don't need to do that. I hope that answers the question.
Can I say, I think we're probably going to have to curtail that, Chris. There seems to be a flurry of questions, but I think we really need to move on now. Okay. Obviously, you've spurred a lot of interest. Okay. And I'm sure that you'd be happy to take
Yeah,
questions,
of course.
additional questions,
Of course.
email. Okay. Is that all right? Chris, once again, that's very kind of you. Okay. Right. Provocative questions there. So I'd like to just wrap things up and just finally by thanking both the speakers once again, Chris and Daniella, and also to actually mention to the wider community that there's the next research seminar on the 22nd of May, hosted by the Population Genomics co-leads. So thank you very much once again for attending, and I'm sure that other people will be prepared to take questions offline by email. Okay, so thank you very much.